So welcome everyone. It's a great pleasure to have you all with us today. And I'm sure some of you are calling in from Vancouver, but other places all around the world. So it's great that you've taken time with us to do this 3-minute thesis today. So welcome to the Faculty of Forestry 3-minute thesis competition heat. My name is Nicholas Coops, and I'm the moderator of the program today, and I'm the head of the Department of Forest Resources Management here at UBC. The 3-minute thesis is actually an Australian invention developed by the University of Queensland in 2008. It was seen as a way of getting graduate students to get into practice of talking about their work very quickly. And actually the 3-minute, I believe, comes from the average period of time that you might be stuck in an elevator when you're talking to someone about your work. So really it's a great opportunity to get in the habit of talking about the research you're doing, why it's important, and what you expect the outcomes to bring in 3 minutes to a non-specialist audience. So you can talk about your work and what you're trying to achieve in a quick and easy way without having to go into too much detail, and keep it at a level that is appropriate for a non-specialist. And that's how the 3-minute thesis design came about. It's been done since 2008 from the University of Queensland, and it's now done all around the world. UBC was one of the first universities to start to do that, and we started to do the competition here in 2011. Different faculties do the competition at UBC, and ultimately there'll be a UBC-wide competition, and from that into a Western Canadian competition, and ultimately a Canadian-wide competition for the 3-minute thesis. So the students that we have talking about their projects today had a series of rules: a single static PowerPoint that they have done themselves and submitted. There's no additional props allowed. They're allowed to speak for 3 minutes on their topic, and the topic is related to the project they're doing here in their graduate studies at UBC. The way we're going to do it today, given we're on a virtual environment, is I will introduce the student and tell you what their topic is with their professor. I will count them down, and then they will have 3 minutes to go and talk about their project. I'm delighted to say we have 3 judges that are on the call today that will be judging the presentations as they see them. They are Professor Lorien Nesbitt, who's an assistant professor in forest resources management, Professor Rob Guy, who's department head in forest and conservation sciences, and Professor Stavros Avramidis, who is the head and professor in the Department of Wood Science here at UBC. So they're going to judge on a couple of criteria. Did they like what they hear? Was it comprehensible? Did it give them a sense of what their research is on? And then their engagement. How did they speak? How did they engage the audience for those 3 minutes around their topic? Now, we appreciate in a virtual environment with Zoom, it's complicated. They're not going to judge your sound quality. They're not going to judge the video quality of the presentation. They're really focusing on what you're saying and how you engage the audience as part of the talk. I'm pleased to say there are cash prizes. So the first prize, $250, second prize, $175, and third prize, $125. And the first and second winners from today will go into the UBC-wide, or go into the UBC process and further into the 3-minute theses. Okay, with those rules explained, we have 10 talks, I believe, today that we're going to go through. And so I will start with the first presentation. So again, I will be introducing the person, counting them down, and then we'll have their 3-minute presentation. And a reminder for everyone to keep their microphones on mute, except for the speaker. So without further ado, let's get underway. So it's a great pleasure to introduce you as our first talker, our first speaker today, Sarah Dickson-Hoyle. Now, Sarah's got an image as part of her 3-minute thesis. It does contain impacts of wildfire on houses and communities, and people might find that image distressing. So just a warning of the image that she has, incredibly powerful for a 3-minute thesis today, but some people might find that distressing if you've had some experiences with wildfire. So Sarah is doing her PhD here at UBC. Her supervisors are Laurie Daniels and Shannon Hagerman. So with that, Sarah, Are you ready?
I'm ready.
I will count you down. 3, 2, 1.
As we reach the end of another long winter, I'd like to take you somewhere warm, to a midsummer day in interior BC. The air shimmers in the heat, warm winds blow up the valley. You can feel the dry grass crackling underfoot. You can't remember the last time it really rained. Then you get a call from your neighbor just down the highway. A grass fire has started. It's moving fast towards your community. There are reports of homes already lost. Within 24 hours, your whole territory is on fire. This was the experience of Secwepemc First Nations during BC's record-breaking fire season in 2017. While in Vancouver, we were choking down smoke, the Elephant Hill fire, depicted here, ripped through Secwepemc territory, burning almost 200,000 hectares, destroying homes, and forcing evacuations of thousands of people. As the smoke settled, government and communities faced an even greater challenge of how to recover this devastated landscape, how to prepare communities and ecosystems for future fire events, and how to ensure that First Nations jointly led this process. My research addresses these challenges. For 2 years, I've been deeply engaged with Secwepemc communities as they navigate this joint process of wildfire recovery. Through 3 studies, I'm learning how these communities and their territories are recovering from this fire. With community staff, we're monitoring forest regeneration and impacts to traditional use plants. With elders, we'll document memories of landscape change to guide restoration. And through interviews with staff and leaders from communities in the province, I'm documenting lessons learned to inform ongoing recovery and future fire response. Interview data reveal a partnership catalyzed by First Nations leaders and government stated commitment to reconciliation. How partners discovered shared values, prioritizing cultural heritage and ecosystem resilience over trees and timber. Wildfire recovery is about healing the land, but my findings show it's about so much more. That the greatest value people see from this new approach is the relationships that have been built between First Nations in the province and between communities themselves. But while government may see this as a model for First Nations collaboration, for many community members, it's just the first step on a long path toward true co-management. This means involving First Nations from the start, well before the first spark of fire in their territory. It means greater engagement from fire agencies, being at the table for decision-making, and reintroducing fire to the land. As we reach the end of another long winter, the time for this change is now. With my PhD, I aim to bring this change forward.
Very good. Thank you, Sarah. Very nice job. A very good start. So thank you very much, Sarah, for doing that. Okay. Number 2. I'm pleased to introduce Samuel Grubinger. Samuel's topic is enhancing phenotyping of conifers for climate changes using drone-based remote sensing, and his supervisor is Nicholas Coops. Samuel, are you ready?
I am ready.
3, 2, 1.
The climate is changing. Are your trees ready? Forests are a powerful weapon in the fight against climate change, but they're also extremely vulnerable to its effects. In much of British Columbia, we expect to see hotter, drier summers on average and wetter winters. But more importantly, extreme weather events in all seasons will become more frequent and less predictable. For populations which have adapted to stable environments, this means trouble. It means that the trees which survive and thrive in future climates will have to be ready for summer drought, winter wet, and unseasonable cold snaps. In BC, all harvested land must be reforested by law, and the province regulates which seed sources can be planted where. The system presents an opportunity to select trees with a genetic basis for climate change readiness. Hundreds of millions of seedlings are planted across the province each year. Most come from tree breeding programs, which maintain orchards of selected populations for reforestation. Breeding programs select individuals based on their phenotype, the measurable physical traits they exhibit, in order to change the population's genotype. But phenotyping of forest trees is a major challenge due to their size and complexity. Traditionally, tree breeders were limited to measuring tree height and diameter, simple metrics which ignore the physiological processes high up in the forest canopy, which indicate growth and stress earlier. In my research, I use imagery collected with drones to phenotype common garden trials of spruce and Douglas-fir populations across BC. I use a technology called digital aerial photogrammetry and produce 3D models of trees to extract structural metrics describing height, crown dimensions, and foliage density. I also use multispectral imagers to calculate reflectance indices, which are indicators of photosynthesis, drought stress, and cold damage. In this image, we see canopy height on the left and a multispectral foliage health index on the right. With these types of data sets, we are better able to detect meaningful differences among individuals. Using statistical modeling, I combine these metrics to quantify relationships between climate and phenotypes, and to determine which populations react most favorably to environmental change. These models can be used to monitor reforestation outcomes, identify highly resilient populations, and inform rapidly evolving policy in British Columbia. Ultimately, this work can contribute to a standardized phenotyping framework, which will allow forest policymakers to analyze data from across North America and better answer the question, which trees should we plant for the forests of the future?
Great, nice work, Samuel. Congratulations. Okay. Please introduce our third speaker for the presentation today. I'm going to go a little bit slowly, allowing our judges to record their notes from the previous talk. Luckily, when we're doing this in person, obviously I can dance around, takes a while to get the screen up, get everyone ready. So I'll fill the uncomfortable silence with words. Okay, so now we've got our third speaker. It's a pleasure to introduce Sheryl Yin. Sheryl's question is, who is the winner of Swiss needle cast lineage fight? She's doing a master's in forestry here in the faculty, and the supervisor is Rashad Hamlin. Sheryl, are you ready?
Yes, I am.
3, 2, 1.
When look at this drawing, you may wonder, who are those three weird guys? Well, they are the structure of sexual reproduction, aka fruiting body, of a fungal pathogen called Swiss needlecast. They may seem small and harmless. However, they are pushing out from the breathing holes of Douglas-fir needles, and eventually they will choke the tree and stop it from growing, which causes not only damage to our beautiful Douglas-fir, but also tens of millions of dollar loss in timber industry. So on the drawing, each of the faces represents a distinct genetic lineage, or you can also call it variant, of Swiss needlecast in Pacific Northwest, namely variant 2, variant 1 coastal, and variant 1 interior. I assume you have heard of the word variants a lot on COVID news lately, right? Just like in virus, some fungal variants are more dangerous than others to human bodies or to plants. My study aims to characterize the three Swiss needlecast variants and infers their relative risk of future spreading. I developed a very sensitive identification tool that amplifies DNA and tells you the exact variants from fruiting bodies, just the bad guy in the drawing. And using this tool, I examined thousands of fruiting bodies from locations across BC and generated an up-to-date lineage variant map. Moreover, I grew 27 individuals from 3 variants under different temperatures and different moisture conditions on artificial media in order to compare their growth performance. As a result, I found variant 1 coastal grew significantly faster than other 2 at all 3 temperatures and 2 out of 4 moisture conditions, indicating that variant 1 coastal is adapting better and can adapt to a wider range of environmental conditions. To sum up, my study provides a molecular identification tool that can be useful for Swiss variant surveillance programs. Besides, my study reveals that one coastal could be the winner of the variant competition under the rapid changing climate. We need to keep our eyes wide open for the variants of pathogens in order to protect our beautiful and valuable Douglas-fir forests. Thank you.
Nicely done, Sheryl. And it's great to see these different images. We've had photographs and graphic images taken from remote sensing and now handheld drawings as well as ways of communicating our science. So I guess that's part of the challenge for the 3-minute thesis people, is to not only get the talk to cover 3 minutes, but get one graphic that's able to capture the work that they do. So some really nice ones so far, and great to have both forest resources management and forest and conservation sciences represented so far in our talks. So nice job. 3 good ones to start with. Okay, so we'll move to our 4th presentation now. So our presenter now is Francois du Toit. Francois is talking about deriving internal crown geometric features of Douglas-fir. Obviously a nice common theme here for our talks so far. Douglas-fir from LiDAR in tree improvement programs. And Francois is here at UBC in the Department of Forest Resources Management. His supervisor is Nicholas Coops. Okay, Francois, are you ready?
I am, yes.
3, 2, 1.
Trees are a very important resource for the economy of British Columbia. The forestry industry contributes billions of dollars to the provincial GDP and accounts for more than 30% of all provincial exports every year. As a result, scientists have been using tree breeding programs to establish high-yield seed sources since the 1950s. These programs don't only grow bigger trees, but also ensure that genetic diversity is maintained while developing sustainable plantation forests. Douglas-fir is one of the most important commercial species in the Pacific Northwest and has been bred to have increased volume over several generations. While successful, very little is known about how these trees are different from regular wild trees. My research focuses on describing the structural differences between wild trees and those that have been genetically improved. In order to increase stem volume, programs aim to change gene frequencies for a few key traits. In Douglas-fir, we select primarily for increased height and diameter at breast height. Currently, these measurements are taken in the field, and they're labor-intensive, expensive, and time-consuming without being particularly informative. Remote sensing can address several of these limitations, as well as supplement other parts of the tree improvement process. LiDAR produces 3-dimensional point clouds, which allow us to measure trees and interpret the structure in a way that goes beyond simple ground measurements. Using these dense 3D point clouds, we can create metrics that describe the vertical complexity of the crown. In my study, we investigated the utility of LiDAR to produce metrics that describe branch attributes such as branch angle, branch length, and branch volume using a point clustering approach. These metrics were chosen as they've been shown to affect the wood quality of the trees. We tested whether these attributes showed differences between trees with different levels of genetic improvement, as well as different planting densities. Our findings agreed well with the manual measurements of branch angle, and we found that the top trees were often quite different at wide spacings, but that these differences dissipated at higher planting density. These findings are encouraging as we try to build links between genetic improvement and how it manifests in nature. These findings are important as wood quality and branch attributes are becoming more important selection criteria as we enter the next generation of improved stock. These new data collection platforms increase the flexibility of when and how we collect data, as well as decreasing the cost. Technological improvements also mean that trees can be analyzed at a higher resolution than was previously possible. The data collection and analysis methods discussed here can be integrated into large-scale programs. We'll be able to efficiently monitor trees, as well as identify new trees, display the attributes associated with larger volumes, thereby increasing the value of forests in British Columbia. Thank you.
Nice job. Exactly 3 minutes. So very nice to hear about this work, and it's great to have Yusri with us because Yusri is actually a co-supervisor on both Francois and Samuel projects. So Great to see some collaboration in the faculty there as well. Switching gears, I guess, from forest wood to perhaps more of the other ecological goods and services that forests can provide. Our next speaker is Spencer Dakin Kuiper. So Spencer is doing his master's here in Forest Resources Management, actually co-supervised by Nicholas Coops and Scott Hinch, looking at the prediction or characterization of salmonid habitat using airborne laser scanning data. Spencer, are you ready?
Yes, ready to go.
3, 2, 1.
Every year in the Pacific Northwest, as summer ends and the days begin to shorten and the leaves start to change color, a great migration occurs. Millions of Pacific salmon start swimming into rivers, beginning the most difficult and the last journey of their lives. As salmon migrate upriver into the headwater streams, they utilize specific habitat features along the way, resting behind log jams before jumping over waterfalls to reach the gravel bed streams that they themselves were born in. The next generation of salmon continue to use these habitat features throughout their life, resting in deep pools and ambushing prey in fast-moving riffles until they're ready to return out into the open ocean. Salmon provide roughly $2 billion annually to British Columbia's economy through commercial and recreational fishing. But their real value lies in the immeasurable ecosystem services they provide, transporting nutrients from the ocean into terrestrial ecosystems, feeding both wildlife and forests. The problem is that these freshwater habitats that the fish are using are disappearing through anthropogenic influences like logging, channel damming, and climate change, ultimately affecting the health of these salmon populations and associated forests. My research is looking at ways to use remote sensing data, primarily airborne laser scanning or LiDAR, to map and measure stream and vegetation attributes important for salmonid species in their freshwater riparian habitats. LiDAR is an acronym for Light Detection and Ranging, a developing technology used by foresters in British Columbia for enhanced forestry inventories. A LiDAR sensor sends laser pulses to the ground and records the amount of time it takes for each pulse to return. Used in conjunction with an accurate GPS, this technology is able to generate accurate three-dimensional point clouds of both vegetation and sub-canopy terrain, as seen in the top left corner of my slide. Using advanced remote sensing techniques on the LiDAR-derived three-dimensional point clouds, we can create a digital elevation model from which we can delineate streams across an entire watershed. Narrowing the focus to the small headwater streams used by juvenile salmonids, we can generate local terrain and vegetation attributes like slope, canopy height, surface roughness, and understory vegetation cover, from which we can directly extract or model important stream attributes for juvenile salmon, like stream width, gradient, in-stream wood, and discrete habitat units like pools, riffles, glides, and cascades, as shown in the image to the right of my slide, as well as riparian vegetation attributes like percent canopy cover and basal area that contribute nutrients directly into streams. Identifying and mapping the freshwater salmonid habitats from LiDAR gives forest managers and conservationists important tools to help protect these keystone species by locating critical habitat and spawning grounds. Thanks.
Great, good job. Thank you very much, Spencer, and a very nice slide there in terms of the images of the work you've been doing. Okay, we'll just give a couple of seconds for our judges to finish recording their notes. Robin, I was going to ask you, Forestry's been doing this for a number of years now. What success have we had in the UBC-wide competitions?
We've done very well with the semi-finals. We usually do quite well. Honestly, off the top of my head, because of my nervous energy, I can't remember if we actually had a UBC winner at the very end. I don't think so.
I was saying one. We had one.
We had one? Okay, well, Gail would know.
And so how do they do it with, I mean, there's lots of faculties, and if we send two, do they have multiple UBC-wide heats?
Yes, so they've had heats UBC-wide all week. Today is the last day for all the heats. They actually had an open heat, so orphan people could go over there too, and they had those on Tuesday and Wednesday. And the semifinals will be on the 12th, and the semifinals will be given notice what to do, and they'll have to record a recording and send it to grad studies, and the 12th will be the semifinals, and then we'll be moved on to the finals.
Great. And I think they actually record previous ones as well. You can actually go to the website and see other work that's going on at UBC. It's a great way to see other people's work in the past for these 3-minute theses.
That is correct.
Great. Okay, so let's move on to our next speaker. It's a pleasure to introduce Rosa Corbet. So Rosa is doing her master's here in the Faculty of Forestry in Wood Science. Supervisors are Chris Gaston and Rob Kozak. I saw Chris on the call, so thanks for coming in and supporting your student, Chris. So this is Rosa, and her topic today is old ways of knowing, new ways of doing. Very nice. Rosa, are you ready?
Yes, I am.
3, 2, 1.
Hi, everybody. I was just wondering if any of you have seen the carved door that is outside of the dean's office. Well, what some of you may not know is that that door was part of a project in 2015 called Opening Doors, where 3 schools came together, the Emily Carr School of Art and Design and the Freda Daisling School of Pacific Northwest Coast Art and co-op here, the Wood Science Building, to have 2 cohorts of students able to carve panel doors and then use the advanced wood science technologies that we have in forestry to scan and then reproduce these doors, making limited edition pieces. So here comes the questions. What's going to happen in this digital age, this technology age, to old forms of artwork like Pacific Northwest Coast art? So I devised 3 questions to research this topic. The first is, what is the relationship between carvers and technology, to understand from their experiences and their opinions? Secondly, how is authenticity perceived by carvers, by the community, and using for carvers who are using this type of technology? And then thirdly, what is the ability for these technologies to increase capacity? What are some setbacks and what are some, essentially, the pros and cons? So I went and interviewed 22 participants from all over British Columbia. And what I found was actually really quite interesting. That first and foremost, technology is a tool, and tools are a means to an end. So most of the carvers didn't actually have an issue with using these advanced science technologies, except for in the case that it was appropriated or used by non-Indigenous carvers or of the like who were trying to reproduce this type of work. The other thing that I found, which was opposite to what I thought at the beginning, was that the experienced and older carvers were actually earlier in adapting and adopting these technologies into their practice. So one of the carvers even told me that it is traditional knowledge and knowing to adapt to things, to adapt to what's available at the time. So I thought that was extremely interesting. The last piece was there was a current of this idea that there's a romanticization around what First Nations carving is and how it can be done. So onlookers and non-Indigenous folks have often this idea that they want the blood, sweat, and tears of a piece of artwork from the artists themselves. But what we are finding is that really that's putting a box around carvers, and that's not exactly how they like to be presented or identified as. So this study is actually helping myth bust the idea of these preconceived notions of what First Nations carving is and how it can be done. And the last question I want to leave you with is, what if AI might be able to create these designs in the future? Thank you.
Great. Nice job, Rosa. And these doors are absolutely spectacular. I have seen them for a number of years now and talked to Chris about them. They are extraordinary. So what a fantastic photo and what a great project. Alice, I don't think my timer is working, is it? Because it still says 2 minutes here. So the timer on the slide doesn't seem to be doing it. But if you look, future people, if you look at Alice, she is holding up the sign in the window for your timer. Don't pay attention to the timer on the slide. Okay, so it's great to have Rosa speak, because now we've had all 3 departments represented. in the 3-minute thesis so far. So great to see graduate students from across the board, no pun intended, coming to give the talk. Okay, people are asking about, well, let you know about how it's going to go today. At the end of the presentation today, normally when we do this live, we hold you back and we have some pizza and then we do the presentations. Given that it's the virtual forum today, the judges will meet after we finish today and they will make their decisions, and then the decisions will be announced by email later on today. So you won't hear immediately who the winners are, but there'll be an announcement later on today that will say that. And Stavros has got a link there in the chat about these stores as well for those interested to hear more about that project. No, not about that, but of something of interest. Okay, so let's move on to our next speaker. Our next speaker is Evan Muise. Evan's topic is monitoring ecological integrity in protected areas using satellite remote sensing. He's in forest resources management and his supervisor is Nicholas Coops. Evan, are you ready?
Yes, I am.
3, so you'll watch Alice for the time. You understand
I've got a timer on my own.
Good to go.
Yep.
3, 2, 1.
The COVID-19 pandemic has dramatically increased the number of people visiting parks and protected areas, making their ecosystem health increasingly important. These protected areas are integral to biodiversity conservation and provide valuable ecosystem services across the globe. In British Columbia, there are over 900 protected areas, which vary in objective from recreation to strict ecological preservation. These parks are frequently managed to promote ecological integrity, which is defined as the tendency of an ecosystem to have its natural components and processes. Ecological integrity is frequently examined in 3 parts. Firstly, ecosystem processes such as carbon cycling and forest disturbances. Second, ecosystem structures such as regional forest proportion and local tree canopy cover. And finally, biodiversity, the proportion and diversity of species in an ecosystem. One of the problems with ecological integrity is finding a cohesive way to monitor it across large park systems, such as the one found in British Columbia. These parks are often found in remote areas where it can be difficult to access them and expensive to conduct ground surveys, preventing the implementation of frequent monitoring for each park in the system. Remote sensing, specifically the Landsat program, provides an excellent solution to this problem as it has global spatial coverage at a frequent return time over a long time series. Through recently produced yearly datasets, it becomes possible to monitor processes such as disturbances in biomass, landscape structure through land cover classifications, and derive fragmentation statistics, and even the forest structure of an individual pixel, produced by combining the Landsat spectral data with airborne laser scanning forest structure attributes. Furthermore, biodiversity can be monitored using forest fragmentation metrics as an indicator of habitat availability. By using these new Landsat-derived datasets to examine components of ecological integrity, we can then compare protected areas to their surrounding environments and ecozones. For example, in the Garibaldi complex, shown in the bottom right, chosen due to its close proximity to Vancouver, forested area is actually much less common in the protected area than it is the surrounding regions due to the elevation gradient within the protected area. This leads to forest canopy cover and height being reduced and the region having less biomass. Anthropogenic disturbances are less common in these protected areas, while natural disturbances such as fire are occurring at similar rates. While I've given you a single park example here, it becomes much more powerful once you apply it to all of the suitable parks of the project, which is part of my master's thesis. Report cards for each park are being generated, providing the necessary information ASAP as possible to BC Parks Management, which will aid in their monitoring programs. Furthermore, examining trends in these variables, both inside and outside the protected area boundaries, provides an indicator of health for these ecosystems. By combining these three components, remote sensing can be used to generate more holistic data for protected area systems and determine if and how well they're protecting healthy ecosystems across British Columbia. This research is going to help inform BC Parks' planning and monitoring program in the future by analyzing trends found over the past 35 years. Thank you.
Nice work, Evan. Very interesting project. And I think there's over 500 parks or so that you're going to look as part of that work. So that's very nice. Okay. Moving from a master's student to a PhD project now out of Forest and Conservation Sciences. Laura's doing her PhD with Rob Guy, one of the plant physiologists we have within the faculty. And her topic is taking a phytobiome approach, linking a plant, associated organisms, and the environment. So Laura, are you ready?
Yes, thank you.
3, 2, 1.
Hello, everyone. Usually when people see a plant, they just see a plant. But have you ever thought of a plant as its own ecosystem or biome? To do that, you actually have to think about things in a way that goes from reductionist to holism, which really requires thinking inside and outside of the box. So why do I care about this? Well, for many years, it feels like now, I've been involved with my PhD project, which involves going from a very small scale to a much larger scale. So let me go into more detail. So for example, one of the things that I look at in my PhD is I've done a growth chamber experiment where I looked at how amending the soil from clear cuts affects the above and the below ground microbiome of the plant. So I was able to find that what you adjust in the soil, such as refugia plants, can actually affect not only the below ground community of microorganisms, but also the microorganisms found on shoots. So one of the things that I also looked at in my PhD and I'm working on right now is a field experiment, which is at a larger scale at Malcolm Knapp Research Forest. This involves looking at nitrogen deposition and also climate warming that is simulated by open-top chambers and fertilization to look at more of a community approach, which goes up in complexity. So not only looking at tree seedlings, but how the community of other plants, as well as associated microbial communities in the soil associated with the roots and the shoots, are shifting with these anthropogenic changes. Also, I've added a project into my PhD that looks at a large continental scale phytobiome approach in the continental US. And I was able to get down to a subset of plots which allow us to look at patterns in species richness, which is the number of species of microbes in relation to vegetation types. So why might this matter to people who aren't really that caring about plants? Because I obviously love them. I'm super passionate about plants. Well, by understanding a more complex system for plants, we can better address things like crop disease, forest pathogens, and other things that we used to have a one disease, one pathogen, but that's just not becoming the case. The more complex ways really allow us to solve these deep problems. Thank you so much. Have a great day.
Many thanks, Laura. Nice graphic as well. Makes you think about it when you're talking. So a really nice job of bringing together your talk and your graphic as well. Okay, making good progress through our presentations today, good representations, and I think it's been great how people have been bringing in common themes. I've heard variant and COVID and all sorts of ways of making work really relevant to the general audience. So I think that's been a really nice example of the slides we've seen so far, and I'm sure we'll continue to talk about. Great to see people linking things the public are able to relate to easily enough and bring that to help give our research hooks that we can talk generally about what we're doing. Okay, so pleasure to introduce our next speaker is Rick Nugent. Rick is doing a PhD here in Faculty of Forestry in Forest Resources Management, and his topic is monitoring early regenerative vegetation using drones. Rick, are you ready?
Yes, I'm ready.
3, 2, 1.
Forests dominate many landscapes in Canada, but they are increasingly affected by resource extraction. This goes hand in hand with many challenges, such as landscape fragmentation, erosion issues, as well as invasive species. This leaves us with the responsibility to minimize our impacts on the land. Key for this is ecological restoration, which is a process in which we seek to restore ecosystem functioning towards historic or baseline conditions, leaving behind self-sustainable ecosystems. But this is not as easy as it seems. Ecological restoration is not exactly like baking a cake. We don't know what ingredients we need to use exactly. We don't know how long to bake it, and how is the end situation actually going to look like. Ecosystems are never static. They're always changing throughout time and space, but within certain thresholds. We focus on pipeline restoration sites in Northwest Alberta. The key ingredient here is leaving soil layers intact, including all the roots, all the nutrient pools, as well as seed beds. But still, this is basis for rapid natural regeneration. But still, this does not guarantee that vegetation is coming back in compositions and abundancy as desired or as on reference sites. For example, water tables can be high or competing vegetation can be present, limiting growth of conifers seedlings. So we need tools that tell us where regeneration is actually matching what we are expecting and where issues arise. This can help restoration professionals selecting locations where we need to take mitigation measures. We've acquired many overlapping images using drones on the restoration sites. From these images, we can reconstruct vegetation in 3D, as seen on the bottom of the slide. These images also include spectral information, and from this combined, we can map vegetation structures, we can map vegetation types, and even specific species. And we can also identify hotspots that are high in species richness. Finally, drones enable us to develop reference frameworks at reasonable costs. We developed such a framework, incorporating sites as in similar ecoregions as the restoration sites and at similar stages of successional development. This tells us what kind of vegetation types and what structures are typical at early growth stages. Based on this, we can develop benchmarks for the restoration sites without having to know how the cake is actually looking like at the end. Thank you.
Nice job, Rick. Well done. And perfectly timed, I might add. And great images there as well. I just counted, so we got 10 speakers today. 6 of them were PhDs, 4 of them were masters. Very hard to tell the difference though. So congratulations to the masters and the PhDs for presenting both really interesting topics, but also at a very high level of quality. So very nicely done. It's nice to introduce Adam Wu as our last speaker. Adam is from Jack Sadler's lab, which has been one of the very large and very research-focused labs in wood science that we've had for many years. So it's great to see a representative from that lab participate in the 3-minute thesis again this year. So Adam, welcome. Your topic, from newspaper to biofuel. Tell me all about it. Very nice pun as well, or very nice topic. Adam, are you ready?
Yes, thank you.
3, 2, 1.
It is apparent that the world we're living in is experiencing vast developments in electronic communication, which allow us to collect information and share experiences at any moment using our electronic devices. In fact, the event we're having today is a perfect example of how convenient electronic communication can be. But this also makes me wonder, when is the last time that we sit down and read an actual newspaper? Newspaper is mostly made of mechanical pulp, which can be produced through the mechanical pulping of woody biomass. And here in British Columbia, we used to be the world leader in mechanical pulp production, thanks to our abundant forest resources, as well as the clean and cheap hydroelectricity. However, the drop in newspaper demand has forced many of the mechanical pulp mills to shut down, which means right now we're having a lot of infrastructure that worth billions of dollars just sitting out there and nobody wants to use them. Therefore, my research plans to repurpose the existing infrastructure and expertise from the mechanical pulping industry, with the focus on adapting the mechanical pulping technique into a front-end method for the biorefinery process. This is the process where microorganisms such as enzymes and yeast are used to convert the biomass into green bioproducts to displace the conventional fossil fuel-based products like gasoline and diesels. It was found in my research that with the help of some mild chemical treatments, such as the use of oxygen to oxidize some of the components of the biomass, the same technique that increases the surface area of biomass to make strong mechanical pulp can also make it easier for the enzymes to get into the biomass and allows them to break down the biomass into sugars much more efficiently. Rather than fermenting these sugars into a bottle of wine, like what people normally do, we decided to make something better. In our approach, these sugars are fermented into biofuel that can fuel cars, ships, and airplanes as a way to better decarbonize the transportation sector and fight against climate change. Thank you.
Very nice job, Adam, and thank you. I was wondering where the bottles of wine were going to come into this. I thought they might have been there by accident. So glad to see that come through right at the end. A great topic, really important work, and a nice slide as well. Okay, so that's the 10 presentations that we've heard for today. So many thanks to the supervisors that support these students through their programs, and thanks for many of them who have joined the call today. It's also a great pleasure for me to acknowledge the huge work that our UBC faculty, UBC Forestry Graduate Studies team does. So Robin, well, Yusri is the Associate Dean of Graduate Studies, but the Associate Dean doesn't really do that much. It's really the people beneath the Associate Dean, for example, in particular, Gail, Robin, and Alice, that really support our students so significantly through their programs. And I'm sure many of you have had emails throughout your entire program from the team, helping you with your program, any questions you have, any questions about your funding or anything like that, your registration in courses is all answered incredibly quickly. And many graduate students in forestry don't realize how lucky we have it to have such a wonderful team of people that are always so ready to answer all the questions we have. And that's not only from a student point of view, but also from a faculty point of view. Whenever I've got a problem, they're always so quick to answer and are always so happy to do it. So it is an opportunity for us to recognize in these events that they've put together just how great they are for supporting all of our students as well. I know the students, some of you may have regretted saying you would do this a week ago when you started pulling the slide together, but I think it's really valuable. And it actually makes me really proud as a head of department around the quality of the research that we do and the range of research we do. So I want to thank our 3 judges, Robin, Stavros, and Rob, for having to decide which of those 3 out of the 10 are the best. A very hard decision in my mind. Thanks to the team for pulling it all together. And most importantly, thanks to the students for doing high quality research that matters to people and are prepared to come and do that and present their work virtually to a wider audience. So with that, Robin, you awkwardly started us at the beginning with inane chatter. Is there anything you'd like to say to finish us off?
I am not a judge, nor a cat, but Dr. Lorien Nesbitt is one of the judges. I know my name is in the back of your head from all the chatter that's today. But thank you very much, Nicholas, and thank you for all the kind words, and our grad team appreciates that. And I appreciate all the wonderful, wonderful presentations. Well done.
Yeah, I thought I said Lorien's name. Did I not say Lorien's name when I said
my
judges?
name? You said
I'm sorry. The 3 judges are Lorien, Stavros, and Rob. So thank you, Lorien. I didn't mean to replace you with Robert.
We do enough times early on, you know.
Loved your cat joke there, Robin.
Well placed there. Okay, so do we all just leave? Is this the plan now?
Everyone leaves except for the judges and Gail and I.
Okay, many thanks, everyone. Great to see you, and I'll see you in person in 2 years' time.
Excellent.